Okay, last lesson of Biology A, tools of inheritance. These are tools that are going to be a geneticist would be able to use to help identify various types of things that are necessary to know about heredity and genetics and give information to families and so forth. So here we go. All right, so in this presentation, review the chromosomes and genes, investigate karyotypes. That's a tool, pedigrees, that are used in predicting inheritance patterns, survey a variety of common human genetic disorders. Those are the things that we'll see in this particular lesson. Okay, so chromosomes. Again, we understand that chromosomes are just literally containers, right? And they hold genes. And the genes that are on chromosome number 1 are the same for me and everybody else in the human race. Same thing for chromosome number 2, and so on and so forth. They contain genes at specific places. So you would expect these blue genes here, these right here, would be the same on there. The chromosome number 1 would always contain this blue gene, and so on and so forth. So these are your body cells, your somatic cells, where we will have those 46 chromosomes divided up into 2 sets, right? We have one set of chromosomes that comes from your mom and one set of chromosomes that come from your dad. That's called your diploid number. So humans are, the diploid number for humans is what they call 2n, or 2 times 23 is 46. All right, we know that the gametes, the egg and the sperm, have only 23 chromosomes. So those types of cells are different than the somatic cells. So they'll only have half the amount of chromosomes, or the N number. We call it monoploid rather than diploid in the somatic cells, right? Okay, tools of inheritance. Homologous chromosomes here contain the same genes. Alleles of the genes might be different. Sometimes pairs might not separate during meiosis properly, which we've talked about with some of those diseases, like Huntington's disease, not Huntington's disease, Turner's disease, and Klinefelter's. Each parent provides one copy of the pair to their offspring. So again, this side here, the purples might be from mom, the greens might be from dad. And we might have, the eye color gene here, there might be brown on this chromosome, but on this chromosome, it might be blue. So they don't have to be the same, but there's the same gene, it is the same trait. Okay, so karyotypes. Karyotypes are when we take the chromosomes and we pair them up. So we take them out of the nucleus, we look at them under a microscope, we pair them up, and we check and see, okay, so chromosome number 1 looks like they're both there. Chromosome number 2 looks like they're both there. If you look at them, this is about the same size as this one, chromosome number 1. Chromosome number 2, the same size. The banding is the same. You would expect these little centromere things, or the spot in the middle of the chromosome, those will be in the same location. All right, if you look at this one, you can see that the chromosome number 23 here, okay, is not the same size. All right, this one is a lot larger than this one. So this is the X chromosome, so why this would be a male karyotype. So in terms of the number of chromosomes, you got 22 times 2 of the somatic chromosomes, and then the sex chromosomes here, we have 2. So 23 pairs seems like a pretty healthy individual in terms of the number of chromosomes. So what is a karyotype? Click on the link in the actual presentation. It's simply a picture of a person's chromosomes. The chromosomes are isolated, stained, and examined under a microscope using chromosomes in the white blood cells. Chromosomes are arranged from biggest to smallest, the last pair being the sex chromosomes. That's the 23rd pair, used to see if there is any abnormalities. So now if you look at this one, right, you can see at the top of the picture, it says Down syndrome karyotype, or trisomy 21. So instead of having a pair of chromosomes, something happened during meiosis where the chromosomes didn't separate properly, and you wind up having an extra 21 chromosome. And because of that, you wind up having something known as Down syndrome, which has a host of different symptoms or abnormalities that an individual has. So This is known as non-disjunction. So chromosomes fail to separate during meiosis, resulting in gametes may have too many or too few. Okay, so Turner syndrome, this one here, you see there's only one X chromosome. So one of the gametes did not have an X, right? And so you wouldn't see two here, you'd only see one. That's a female, if it winds up being a female. All right, Klinefelter syndrome is the opposite, where the gamete wound up having an extra chromosome. Okay, so there might be that both X's here didn't separate, and so the individual winds up having two X's and a Y. It is a male, and we can talk about some of the symptoms that these individuals wind up having. All right, so down 321st chromosomes, trisomy 21, causes lifelong intellectual disability, developmental delays. In some people, it causes health problems. The Turner's, it's a single X chromosome, affects development in females, shorter in stature, could have some ovarian issues, can be incapable of having children. And then there's Klinefelter's, right? This is two X's and a Y. It affects males' physical and cognitive development, produces less testosterone, and a little bit of a delay in puberty. So this is, if you were looking at this karyotype, you go, only one X chromosome. That must be Turner syndrome. Look at this one. Oh, an extra X and a Y, right? Y is significantly smaller, must be Klinefelter syndrome. All right, so here's some other types of abnormalities. This one is a deletion, right? So this piece of this chromosome did not get, it was chopped off, all right? And so it's missing. So the things that were on this, again, most of the time genes work with other genes to produce various things. So without that region, it does create some issues. This is what it looked like in a true karyotype. So if you see this little piece missing, that's gonna raise a flag. All right, so what is Cri-du-Chat syndrome? This is like, it is translated into cries like a cat. All right, chromosome number 5 deletion, small larynx causes respiratory issues, babies cry, sounds like a cat, hyperactivity or aggression. Right, you can take a look at some more things if you click on the links, but that's another type of usage of the karyotype, whether the genes are intact or not. You can have, remember we talked about translocation where another piece would be added on. Instead of deleted, another piece is added on, causes problems too. All right, so another type of tool is the pedigree. All right, pedigrees are used for analysis with existing population, determine inheritance patterns within a family, can look at inheritance over several generations. Carriers at times will be half shaded in. So you'll see this is half shaded. That means it's carrying a trait. It doesn't, and this one, same thing, it's carrying something. All right, here's the little symbols. Typically used, the male is a square, the female is not. If it's not shaded in, they do not have a particular trait. If they are shaded in completely, they have it. All right, so if they have had children or they're married, something like that, then there's a line that attaches a male and a female. You see the number, this set of parents in generation 1, right, had 2 children, a female and a male, okay? Identical twins, you can see, they do it this way, and non-identical twins will be with a little line that way. Okay, so now when we look at these things, we can determine certain things. So if you look here, this parent, this dad, is carrying a trait. This mom is carrying a trait. So if they both produce an offspring and they give that child right here, each of them gave them that trait, now we fully color that individual in. So this could be some sort of a recessive trait, whether they can roll or not roll their tongue, or that's actually the dominant trait. You know, some sort of recessive trait we'll talk about in a second. So this individual would have it. This couple now has 3 children, 2 girls and a male, and they passed on the mom because she has 2 options, 2 forms of that trait. She's definitely passing on 1 to each of her children. Whereas if you look at this one here, not so much. Okay, so now how do we follow the pedigrees in terms of trying to figure out what's going on with the family? So this one here, this individual has the trait, okay? And this individual does not. Now, because this one does not, they will never pass on the trait to their offspring. However, if you look, their daughter has it and their son has it, which means dad passed on the trait to the offspring, and because he only has one copy of it, it must be dominant, right? In order for it to be recessive, each parent's going to have to pass on one of those traits. So here's a recessive, an X-linked recessive trait, right? So this is a chromosome that is on the X. This is a trait that's on the X chromosome. So if you look at this male, right, because it's X-linked, he only has one X. So whatever's on that X, he winds up having. So every one of his, or the options of him passing on this, generally is higher with a male because this female does not have it. So you look, their offspring did not have any. This couple, their daughter married a male. They didn't have any children with the trait. Their son here didn't have any children with the trait, right? But here, okay, they wound up having the individual pass this on. So this daughter, because she has two X chromosomes and it's a dominant trait, she's not showing it, right? Because her other, or it's a recessive trait, her other X chromosome is hiding it, but she passes on one X to her son. Well, the X she passed on had the trait, therefore her son here is going to take on that trait. So that's how an X-linked inheritance impacts the males. Okay, and that will be it for Biology A.